Welcome everyone to session 9, where we'll be speaking about correlation and regression. So quick overview of this lesson, we'll be going over what the Pearson's correlation and um, coefficient of determination is. Then we'll speak a little bit about simple linear regression. We'll also be completing that analysis in the lab in addition to the correlation. And then I'll quickly overview what the simple logistic regression is. I'll just be providing the information about that, but we won't be covering that in our lab session. We're going to just focus on linear regression and correlation. So, quick review here. We have inference, right, and description for two variables. So we usually think of one variable as a response variable, right, or the outcome variable, and the variable that we think of as, for example, our exposure, or we also can call it our predictor, right? Even when we're not really exactly sure if it's necessarily causing that outcome to happen, we say that they're associated, right? But again, when we're thinking of these analyses in our minds, we think of having, a predictor to a variable that, or an outcome that's responding to that predictor. So inference is performed on, right, the response variable. And the response variable is plotted on the y-axis. So we'll look at some examples of what this means together. Don't worry about it if you don't understand what this is yet. Right, so let's say we are looking at two categorical variables, right? The inference, right, the chi-square test is applicable for variables of two or more levels, right, such as like education level, borough, yes or no. So we saw that in our last session, lesson 8. The description, right, the contingency tables. How are we describing our data? We're using to describe our data, we're using these contingency tables, these frequency tables, and here's an example of that down here, right? Another description for us is the odds ratio or the risk ratio, right? For the special case of both binary variables, right? Yes or no variables, or variables that have even more categories than this. So what we do is we get, the description of our variables using the description of our data using the contingency tables that tells us the frequencies, and then we get a description of the risk or the chance of finding your outcome. And then our actual inference is the test, right? The chi-square test. So here we have, right, our continuous response, right? So specifically in our analysis that we'll be looking at in this lesson in our lab is we're going to be looking at a response variable, right? Our outcome variable that is continuous. We're going to be just focusing on, whether that's a continuous variable, and our predictor is going to be categorical variable. So far, like for ANOVA, we looked at, two categorical variables, and we compared them against each other. Now we're looking at an outcome variable that is continuous, but the predictor variable is categorical. So the inference would be the t-test for a binary predictor, right, for ANOVA. Oh, sorry, t-test for a binary predictor, or we could use ANOVA for a categorical predictor. And then the description would be, a graphical description of that would be a nice side-by-side box plot or a bar plot, right? So we looked at this when it came to, I believe, ANOVA, right? We had like predictor variable, we had the categories, and then we had the response, which was continuous. The number of, let's say the response variable is weight or something like that. And then our predictor was the 3 categories of race, for example, right? So we'd have like, did I say weight? I think I said weight or blood pressure, whatever the case is. Those are all continuous response variables. And then the predictor would be categories, say black, white, Hispanic, or whatever the case is. But we can also represent that information, besides a side-by-side box plot, we can use a bar plot for that graphical representation. All right, so in today's topic, regarding inference and description for two variables, we are going to speak about a continuous response variable and a continuous predictor variable now. So let's say we have two continuous, right, variables. We want to use what's known as a scatter plot, right? Because this is the best way to represent, the X and Y point axes. So each data point can be plotted if they're both continuous, and say Let's say we're looking at a good example of two continuous would be, let's say we're looking at, we could say weight and height, right? Let's say we have the weights across the bottom and the heights across the top. So, let's say this person is at this halfway point, right? This point here. So this person has a 50, they're 50 pounds heavy and 50 inches tall. This is just an example, very hypothetical. Um, so this is the kind of graphical representation you would want to use when you have both continuous response, right? Your outcome is continuous and your predictor is also continuous. So the inference method you want to use is you could use a regression analysis to actually um, create that inference, but we'll be going more into depth what that is in a little bit and in our lab. So a good description or a good graphical representation for the two continuous variables, right? Continuous response and continuous predictor is known as a scatter plot. Typically what you want to do is you want to plot along your x-axis, you want to plot your predictor, kind of like we saw with the three groups before earlier, right? The predictor, you want to plot them on the x-axis, and then the y, you want to have your outcome, like whether that outcome is the number of pounds you gained, right? Just hypothetically. So remember, when the scatter plot is going in this direction, it's known to have a positive relationship, and when it's going this way, x and y is said to have a negative relationship. So let's look at some example applications of this, right? So this is a paper regarding cysteine levels in children taking anti-epileptic drugs, right? Drugs that prevent epilepsies. And this was a randomized controlled trial. So what I wanted to point out here is they did a correlation, right? Their statistical method to test the cysteine levels. And let's read what this says. It says, this was a double-blinded randomized controlled trial conducted in the Institute of Maternal and Child Health in India. And there was a total of 60 children recruited for this randomized controlled trial. 48 were enrolled. And then of these children, 32 were assigned to the experimental group and 16 to the control group. So the baseline data collection of this, what is this, homocysteine levels, I believe this is, right? Homocysteine, THCY, estimation were done. And let's see, what 1 mg folic acid tablet was given to the experimental group and a placebo tablet to the control group for 30 days. The levels were re-estimated after 1 month of follow-up. Statistical significance was tested by using a chi-square test and paired and unpaired t-tests, right? So we learned a little bit about that in lessons 7 and 8. So the correlation, right, to see whether or not the levels was correlated, I guess, by the tablet. The correlation was tested by using the Pearson correlation test, and p-value was less than 0.05, was taken as the cutoff for statistical significance. So I just wanted to point that out. If you guys are interested in learning more about this paper, if you feel free to follow up with, just probably Google it or look on PubMed, right? NCBI PubMed has a lot of research articles, peer-reviewed research information there that's very reliable. And if you have any more questions about this, feel free to reach out. But what I want to show you is how relevant what we're talking about in this lesson and in previous lessons are to research in general. So scatter plots and correlation go hand in hand, right? A scatter plot or a scatter diagram is used to visualize the relationship between the two variables in detail. Like, as I mentioned, you want to plot your predictor along the x, your outcome or response variable along the y. Correlation is used to quantify the strength of the linear relationship between the two variables. So you're only concerned with the linear relationship here. There's no causal effect implied, meaning you don't necessarily want to use a correlation as an inferential statistical technique because it's just showing you how associated the relationship between your two variables are, your predictor and your response or outcome variable, but not necessarily saying, X causes Y. In order to do that kind of inference, again, you want to just make sure you're using really controlled settings, such as a randomized control trial like they did in that example I showed before. And then they can actually say, hey, this causes this. Otherwise, we can point out associations, but we have no way to necessarily statistically prove that it causes, X causes Y. So when we're calculating the Pearson's correlation coefficient, right? First, what we want to do is probably look at it using that scatter plot. And this is the formula for what the Pearson's correlation coefficient looks like, right? So I won't ever require you to calculate this by hand because SPSS will do it for you. And I've actually never had to calculate this by hand because I usually use a statistical software to do so. But if you're interested, here's what the formula looks like for a correlation. And you're basically relating one continuous variable to another, like to show how associated they are. And the value that gives you exactly whether that association is close or not is called R. All right, and we'll learn a little bit more about R in lab. So here's some examples. We have a linear relationship here. This is an example of a linear relationship because, as you can see, from this point to this point, you see like a steady incline. If I drew a line plot along with, like if I drew a combined plot and drew a line of the relationship that I see between X and Y, it will be a straight line across this way.